A new approach to fighting cancer could lie within our own fat cells. Because our own fat cells could indeed fight cancer, not through a direct attack, but by simply starving tumors. What sounds like science fiction is now a tangible reality, thanks to a groundbreaking study published in Nature Biotechnology. This naturally raises the question, how exactly is this connected to cancer?
That brings us directly to the heart of the problem. For over a century, we've known about the insatiable hunger of cancer cells. The German physiologist Otto Warburg discovered that tumor cells consume glucose 4 times faster than normal cells, even with sufficient oxygen. They are true metabolic machines, ruthlessly hijacking resources.
For a long time, the hope was to combat cancer through general starvation, for example, through diets or fasting. But reality showed cancer cells are survival artists. They suffer less than healthy tissue, meaning you would literally starve before the tumor does. A systemic approach hits its limits here.
But precisely where traditional approaches fail, a new hope began to sprout, specifically with the discovery of active brown fat in adults. Unlike white fat, which stores energy, brown fat burns calories to generate heat. It's a true energy consumer, efficiently using glucose and fats.
Fascinating that our own body possesses such power plants. Researchers at the Karolinska Institute then also discovered when brown fat is activated by mild cold, it aggressively competes with tumors for glucose. In mice, this led to an impressive 80% inhibition of tumor growth and doubled survival rates. Initial human studies also showed reduced glucose uptake in tumors. So the idea was there, but practical implementation remained a challenge.
Exactly, because who wants to live in a refrigerator for weeks to fight cancer? The groundbreaking solution came from scientists at the University of California, San Francisco. They used CRISPR technology to genetically modify white fat cells. They reactivated a key gene called UCP1, which is normally found in brown fat. The result? So-called beige fat cells, or bright cells, that possess the same energy-consuming property, all without the need for cold.
And the results from these beige fat cells are spectacular. These modified fat organoids were implanted next to tumors. The result? An over 50% reduction in tumor volume for breast, pancreatic, colon, and prostate cancers. The fat cells specifically deprive the tumors of nutrients, starving them out and thus inhibiting their growth.
That's truly impressive. The potential of adipose metabolic therapy, or AMT for short, is enormous. Fat cells are easy to extract, multiply in the lab, and reimplant. They cause hardly any immune reactions. This allows for personalized therapy where the body's own cells become a weapon against cancer. In the future, even controllable systems could be used which can be activated or removed by medication.
This research reveals a new, pioneering path, transforming a seemingly simple tissue into a precise metabolic fighter against cancer.
It's a promising approach that underscores how a deep understanding of biology can lead to revolutionary solutions for some of the greatest medical challenges.
We hope we've been able to give you an exciting insight into the future of cancer therapy.
Thank you for watching. Stay tuned for more insights into the science that will change our lives. And as always, we wish everyone a long, healthy, and happy life. Until next time.